Lab 2, Introduction to the Microscope. In today's lab, we're going to complete 3 exercises. In the first exercise, I'm going to give you an overview of the parts and functioning of the microscopes that are used in the Biology lab. In part 2, we're going to learn 2 different ways to calculate the size of specimens that can be viewed under a microscope. And in part 3, we're going to talk about how to do scientific diagrams. For the microscope that we're using in today's lab, I have 2 video clips that I'm going to play here right away. The first video clip is an introduction to the parts and functioning of the microscope. And the second clip is how to prepare a wet mount. I'm going to play those for you right now. Today I'm going to introduce you to the microscopes that we use here in the Biology 107 lab. In fact, you can see a whole bunch of the microscopes that we use in the biology lab on that shelf behind with the blue coverings. So here is our microscope. It is called a Zeiss Primo Star microscope. And what I want to do is introduce you to some of the parts and the functioning of the microscope. So let's take a look at this thing. So up here we have the ocular lenses, or the eyepieces. In fact, these microscopes have 2 for binocular vision, and what that can do is actually give you a bit of a better view, a little bit of a stereoscopic view. 2 eyes are better than 1. You can see these eyepieces, they will actually move apart or closer together. Kind of just depends on what's comfortable for you and your eyes. So these here are a 10 times magnification. If we move down the tube here, down over here we have the objective lenses. So let's go in for a little bit of a closer look. So the objective lenses, you can see that there's 4 different ones, and let's take a look at this one here. This is the 4 times objective lens, so this is the smallest objective lens, and you can see the numbers written on here. It says 4 times, and over here that's the numerical aperture, so it says 0.10. And that's important for figuring out what your theoretical resolution is. So in this case, if we have a 10 times ocular lens and a 4 times objective lens, then 10 times 4 is 40, and that would be what you're viewing the specimen at. So over here, this is the 10 times objective lens. This one with the blue on it is the 40 times objective lens. That one's pretty commonly used in Biology 107 because it's really good for looking at eukaryotic cells. And then lastly, we have this white one. This is the 100 times oil immersion lens, and we're going to use that in Lab 4 when we look at bacterial specimens. So a little bit more on that later. So just below the objective lenses is the stage. And you can see there's a little bit of a lever here, and what I can do is I can put a microscope slide in that stage. So let me do that for you right now. And I'm going to just put that microscope slide in. Unfortunately, this needs 2 hands. So there I just put a microscope slide in, and what you can see is I have prepared a wet mount. And we'll talk about how I prepared that in a minute or 2. So moving down, underneath here, we have the light source is basically in this base here. And in order to turn on the light source, we have to first plug this thing in. So there's a plug at the back. I can plug it in right here. And over on this side is the on-off switch right here. So you can see the on-off switch. These lights turn on and show you what setting it's at. You can turn it down or turn it up. If I take a look at the actual specimen, you can see the light is, you can see it's getting bright or dim. And it's really just a matter of preference in terms of what you want to see your specimen best at. Sometimes with the larger lenses, you need a lot more light. With the smaller lenses, you need a lot less. So in order to focus, what I'm going to do is I'm going to put this actually at the lowest lens here, and this leads us to these knobs here and these knobs here. So these knobs here are actually the stage adjust knobs. So watch what happens to my slide when I turn these. So one of them moves the slide horizontally, the other moves it up and down. So the whole idea here is to center the specimen basically on the light source. Down here, these are the focus knobs. So in order to focus, what I need to do is turn the one on the outside. So there's actually two knobs, one on the outside and then there's the one on the inside here. So watch what happens when I turn the coarse adjust knob, which is the one on the outside. You can see the stage, it goes up and down. And so this will focus and get kind of a rough focus. And then once you're kind of almost focused, you're going to move the inside knob here, which is the fine focus, and that also moves the stage up and down, but just kind of at a very small degree so that you can focus on everything a little bit sharper. So that is the PrimoStar microscope. So hopefully you enjoyed my tour of the microscope, and we'll take a look at the rest of the lab exercises that we have for today. What I'm going to do right now is make a wet mount. So I have all the supplies that I need. First thing I'm going to need is a microscope slide. So I will get one of those. There we go. Next thing that I need is my specimen. So this is the specimen that we're going to be using. It's an aquatic plant called Elodea. Now Elodea is really nice because the leaves are very, very skinny. They're actually only 1 or 2 layers of cells thick. So they're very nice to view on the microscope slide. So what I'm going to do is try to find a nice looking leaf, something that's still green and is going to hopefully look nice under the microscope. So add that to my slide. Uh-oh. There we go. Okay. So this is a wet mount, so I need a little bit of water. So I'll add a drop or 2 of water just to keep things fresh. And then the last thing we need is something called a cover slip. And a cover slip basically looks like this. It's a small, skinny piece of glass that's just going to keep my specimen nice and flat and keep the water on it. And so there we go. I'm done making a wet mount. Okay, so now that you are oriented to the parts and functioning of the compound light microscope, I want you to go to your lab assignment and you can fill in this table. So what you can do for each of the objective lenses, or at least the 10, 40, and 100 times, is fill in this column here, total magnification. You can see the formula is down at the bottom. The numerical aperture is found on each of the lenses. There's a photo of those in the lab manual, and you can also calculate the theoretical resolution. This particular table is going to be very useful to you. In particular, these 2 columns here are going to be essential for determining the size of a specimen. We're going to use those in today's lab, and you're going to use them in future labs as well. So keep in mind you may have to refer back to this table several times over the semester. So our second exercise today is calculating the size of a specimen. So what if you have a cell and you want to figure out how big it is? We are going to be looking at 2 methods. One is the proportion of the field method, and the second is using an ocular micrometer. So let's first talk about how to calculate the size of a specimen using the proportion of the field of view. So if you look at a specimen under the different objective lenses, it's going to look different sizes. So let's take a look at an example. So in this hypothetical cell, at the smaller objective lens, the 10 times, it looks tiny. Under the 40 times, it looks a little bigger. Under the 100 times, it's bigger yet. So what I'm trying to show you is that you have a field of view. When you look under a microscope, it looks like a big circle of light. And if your specimen's on there, it's going to look a certain size. If you look at table 2-1, I've given you some information about how big that field of view is. So the 10 times objective, the field of view is 2 millimeters, the 40 times it's 0.5 millimeters, and the 100 times it's 0.2 millimeters. Now when we measure cells, usually we measure cells in micrometers. So 1,000 millimeters is 1 micrometer. So if we want to convert from millimeters to micrometers, we multiply by 1,000. So 2 millimeters is 2,000 micrometers. 0.5 millimeters is 500 micrometers, and 0.2 millimeters is 200 micrometers. Let's take a look at an example to show you how we can figure out the size of a specimen using this particular information. So if you take a look at this, here is the cell under the 100 times. So when I'm talking about the proportion of the field, the whole idea here is figuring out what fraction of the field that cell takes up. So that's a pretty big cell, and I'm estimating it's about half of the field. So in that case, 1/2 is the proportion, and you can make that into a decimal, which is 0.5. And then basically, I also have the other information, which is the size of the field, so 0.2 millimeters. To get the size of the cell, I just multiply the two, the proportion, the fraction, times the diameter of the field. So that would be, of course, 0.5 times 0.2 millimeters. That gives me 10 millimeters. And when I convert that into micrometers, I multiply it by 1,000 and I get 100 micrometers for that cell. Let's take a look at the same cell under the 40 times magnification. So in this case here, it's a little harder to estimate exactly what proportion of the field that is. So the way I like to do it is to think about if I were to stack these cells side by side, how many would fit across that diameter. So in this case, I'm going to estimate 1, 2, 3, 4, and 5 cells would fit across the diameter of that field. So in this case, the proportion is 1/5, or 0.20. So it's the same calculation. I take the diameter of the field. In this case, the diameter is a little different. It's 0.5 millimeters. And I multiply those two numbers. So 1/5 of 0.5 is, of course, 0.1 millimeters. So in micrometers, I'll multiply that by 1,000, and I get 1,000 micrometers. So different numbers, I came to the same conclusion, the same size, and that's always good news when your numbers match up. Here are some images of some real cells. These are some epidermal cells. This is some sort of preparation, and you can see the cells under 10 times, 40 times, and 100 times. Now, it's really hard to see the cells in any decent amount of detail in the 10 times, so I'm just going to skip over that and go right to the 40 times. Let's take a look at that sample under the 40 times magnification to see if we can estimate the size of that cell. So there is one cell I'm going to look at, and if I'm looking at the proportion of the field of that, I'm going to take a little bit of a guess. This is where the estimation comes in. I'm kind of using my finger and a pencil to estimate, and I figure about 12 of those cells would fit across the diameter of that field. So my proportion in that case is 1/12, which is 0.083. So the diameter of the field is 0.5 millimeters. I multiply those two numbers, and 1/12 times 0.5, and I get 0.0417 millimeters. I convert that to micrometers by multiplying by 1,000, and I get about 42 micrometers. So that is my estimation using this particular method of the size of that cell. Let's take a look now at a second way to measure the size of a specimen. This is using something called an ocular micrometer. So an ocular micrometer is actually a much more accurate way to measure the size of a specimen because we are actually measuring it directly. So what is an ocular micrometer? It's basically a glass disc. It looks something like this, and it is built right into the eyepiece of the microscope. So if you zoom in on the ocular micrometer, it looks something like this. It's basically a little graduated scale, and we can use that directly looking at a specimen kind of like a ruler. So the ocular micrometer is in the eyepiece, so it looks the same no matter what objective that you look under. So the actual values between each of the measurements for the ocular micrometer is going to be different depending on which objective you're actually looking under. So if you see this under the 10 times, 40 times, 100 times, it looks like the same size. But remember, when we look at our cells, we're actually zooming in on the actual cell. So the scale for the ocular micrometer is different for each objective lens, and that actual scale, that conversion factor, can be found in table 2-1. So make sure you always look at 2-1 because this is going to help us to figure out the size of your specimens. Let's take a look at some examples. In our first example, I'm going to look at the 10 times, and notice I've zoomed in a little bit just to make it easier to see the numbers. And so in this case here, there's my cell, and each of those divisions is considered one division, so my cell is 10 divisions. And so we call those 10 ocular divisions. And then if we look at table 2-1, the number of micrometers per division for the 10 times lens is 10 micrometers per division. So in this case, it's a simple multiplication, 10 times 10. So 10 times 10 micrometers per division is, of course, 100 micrometers for that particular cell. Let's take a look at the 40 times lens. So notice in this case here, the cell is quite a bit bigger. I've also zoomed in just to make it a little bit easier to see the numbers. So in this case here, it's a little bit beyond the scale, but so I'm estimating. In this case here, I'm estimating that that particular cell is 40 divisions long. So the number of micrometers per division for the 40 times is 2.5 micrometers per division. So same like before, I multiply those two numbers. 40 times 2.5 micrometers per division is 100 micrometers. So I'm getting the same number, and I'm getting the same number actually I found when I was using the proportion of field method as well. And so this is always good, like I said, when your numbers do match up, and so I'm pretty happy with that. Let's take a look now at this particular sample, these epithelial cells. Let's zoom in, and there we go. So there's our cell, and there's our ocular micrometer. So I'm guessing, looking at that, it's going from about the 5 to the 22. So that would be 17 divisions of the ocular micrometer. So the number of micrometers per division for the 40 times is just like the last example, 2.5 micrometers per division. So we multiply those two numbers, 17 times 2.5 micrometers per division is 42.5 micrometers. So that's matching up really well with the number that we calculated before using the proportional field. So again, it's good to see that our numbers do add up. For the Lab 2 assignment, we're going to take a look at cells from Elodea. So Elodea is an aquatic plant. A lot of people actually have it in their fish aquariums. I think the common name is something like the American or Canadian pondweed or something like that. It's actually a really nice plant. It gives lots of oxygen for the fish. So as I showed you in the previous video clip, I made a wet mount of this and took some pictures of it. So here are some pictures under the microscope. And if you take a look at that, there is an ocular micrometer on there, and I'm showing the proportion of the field in the big circle. In case you're wondering, that there is one cell, and I'm going to zoom in on that cell just to talk about what it is that you need to do for this assignment. So by the way, if you're wondering, those little round things are chloroplasts. That's actually a pretty nice picture, and you can see them quite well in that image. So what you need to do is calculate both the length and the width of one cell. And you're going to use the methods that we discussed already, so proportion of field and the ocular micrometer methods, and you're going to compare those results. So for your assignment, you are going to show the calculations for those measurements, and that's going to be submitted as part of your assignment. In the last exercise of today, we're going to be looking at these prepared slides here. So these have been prepared by a scientific supply company. These are onion root tips. So what they've done is they've stained these cells, and you can very clearly see the DNA. And we use onion root tips because you can see the various stages of mitosis. So they're very, very nice preparation. Unfortunately, the preparation wasn't quite big enough to get the full field, so I've put some gray in the background. So make sure when you're using the proportion of the field method to measure the size of your cells, you go all the way out to the large circle. So what you're going to do for this last exercise, besides measuring the size of some cells, is you're going to do a scientific diagram. So I'm not going to go over all the instructions for a scientific diagram right now, but what I do want you to do is take a look at page 212 and also refer to Appendix C for some examples and for some instructions on how to do a scientific diagram. I'll show you a brief example here in a moment of what to do or what not to do and how a scientific diagram works. But do make sure you read these instructions carefully. So here are a couple of examples of some scientific diagrams. and you can see the little caption. It says, which is better? I'm hoping it's really obvious here which is better. The one on the left is far superior, and it's following the instructions that I've given you. So first of all, for example, there's no shading. It has nice solid lines. The diagram itself is on the left. The labels are on the right. A ruler was used to show what the labels are indicating. The one on the right, it's small. We have shaded areas, which is not good. Some of the labels aren't very specific. So the one on the left is far superior. Something else to keep in mind for your diagrams is that they're going to have a title. And so the title is going to have a number. It's going to be figure something. So figure 1 or figure 2, or could be figure 3-1, but it's going to be labeled with a number. And then it's going to have a title that includes what the slide was. So in that case, the slide is onion root tips, but you're going to use the binomial name for onion. So what you're going to have to do is figure out what that is. You'll have to maybe use some Google to figure out what the binomial name of onion is. And of course, that binomial name will be in italics. So prepared slide of onion, whatever the binomial name is, and viewed under whatever objective lens it was viewed under. So something relatively descriptive. Lastly, in Biology 107, we need to have the magnification of your drawing. So I haven't showed you how to do that yet, and I'm going to show you right now how to calculate the magnification of a drawing. So let's do that. Let's show you how to calculate the magnification of this drawing in particular. There is the formula for calculating magnification of a drawing. So the size of the drawing divided by the actual size of a specimen. So how do we get the size of a drawing? Pretty simple. You just get yourself a ruler. So you can measure that. You can measure it in centimeters or millimeters. In this case here, I'm measuring 3.4 centimeters. And how big is the specimen? Well, that was what we did in the previous exercise. So if you remember that one cell, epithelial cell, that we measured, it was 42.5 micrometers. At this point, it's very, very, very important to know that these have to be in the same units to do this calculation. You can't divide a centimeter by a micrometer. You have to convert them both to the same units. So I could convert everything to centimeters or everything to micrometers. I find it easiest personally to convert everything to millimeters. So 3.4 centimeters is 34 millimeters, and 42.5 micrometers, I divide that by 1,000 to get 0.0425 millimeters. So in this case here, the answer is 800 times is my magnification. So this calculation and this final value of 800 should be with your scientific drawing for all Biology 107 scientific drawings. We're now at the end of Lab 2. So this means you have an assignment due for me. So this assignment is a Lab 1-2 short report and also a Lab 3 pre-lab assignment. This is due 1 week after the end of completion of Lab 2. Please check your lab schedule for the exact due date. Here's the actual lab assignment. You can see it has questions from Lab 1, questions from Lab 2, and of course the Lab 3 pre-lab assignment, which is really just the part of the Lab 1 library assignment. So what you can do is answer all of these questions in a single Word or PDF document, and then you're going to submit it on Moodle. So I do prefer a PDF document, but it's not that hard for me to convert from Word. And if you could, it would be great if you could name that document with the course name, the lab number, and of course your name, which may be the most important item to add to that file name. By the way, this particular assignment is going to actually have 2 documents you're going to submit. So one will be the answers to all of these questions. The second document will be a PDF of the journal article that you find for question number 7 here. So if you have any questions, please contact me, and I'll be happy to go through any of these questions, explain any part of the labs to you that you do not understand. And good luck, and we will see you in lecture.